Hello students, in this video I am going to tell you about the fertilization and implantation. In last video we saw about the menstrual cycle and their all phases. So in this I am going to start from fertilization. So first thing, what is fertilization? So simply it is the fusion of gametes, and in this process the fusion of two types of gametes, that is spermatozoa and ovum, or simply sperm and ovum or ova. So they fuse together and form a diploid zygote. So that is 2n. So fertilization occurs in human beings inside ampulla, isthmus, junction, part of oviduct or fallopian tube. So point to be remember that ampulla, isthmus, junction, part of oviduct or fallopian tube. This is also called as ampullary isthmic junction. So I have divided these fertilization and implantation fertilization into so many sub parts. Like first is the arrival of ovum. So in this what happened, that ovum in secondary oocyte state is released from ovary during ovulation, as you have seen that in menstrual phases, that ovulation, that it is in the you can say middle of the menstrual cycle, almost at the 14th day. What happened, ovum in secondary oocyte state is released from ovary, and due to slow due to slow ciliary movement, the released ovum, slow ciliary uh, current or movement, the released ovum passes into nearby oviduct through that fimbriae. I think you remember the structure, that fimbriae, or this also called as fimbriae funnel or infundibulum part. So and ovum reaches to where, ampulla, isthmus, junction. So the first part is arrival of ovum and how the ovum reaches to ampullary isthmic junction due to their ciliary movement at the part of fimbriae or infundibulum. And do you know that there is so much of ciliated epithelia and so much of cilia is there, and it takes 12 to 24 hours for the ovum to reach this part. or you can say at the site of fertilization. Then the second is insemination. So what happened in this in insemination? It is the process of deposition of sperm in the genital tract of female. Deposition of sperm in the genital tract of female during copulation or coitus. So the inseminating material is actually a viscous fluid, and that is called as semen, and the quantity is about 3 to 4 ml per ejaculation, and it contains almost 80 to 100 million of spermatozoa per ml per ml. And after passing into vagina, semen is converted into coagulum. This also I have explained earlier. And this semen will dissolve after 15 to 30 minutes. After 15 to 30 minutes, and it frees the sperm, and which become motile. So a proper pH is also responsible for this, and that proper pH of semen is provided by the ingredients of semen. This also I explained earlier. Then the third step is motility of sperm. So sperm can swim in a liquid medium, which is provided by the secretion of genital tract. As you know that cervix releases some mucus, and cervix also develops a mucus string of mucus. For what? For guiding the sperm, guiding the sperm. And there are so many constituents of semen, in which there is also constituent known as prostaglandins, which is present in semen, and oxytocin. Oxytocin is released during orgasm of female. So it brings movement of genital tract of the female. Movement of the genital tract of the female, and this result in some of the sperm reaching the fertilization site, that is at ampulla. And normally it takes 5 to 6 hours for the sperm to reach the site of fertilization. So ampullary ischemic junction. And how much time? 5 to 6 hours. And they can remain active for 24 hours. Then next is this one is third, and then fourth. Fourth is capacitation of sperm. This is very much important. Capacitation of sperm. It is the phenomenon of sperm activation. Sperm activation, by which the sperm develops the ability to fertilize ova. Sperm activation means ability to fertilize ova. So the factors present over the sperm which do not allow immediate capacitation are called decapacitation factors. And what are these? There are so many factors which do not allow sperm mobility uh, immediately. So capacitation develops in the female genital tract by the following process. So there are so many things, so many factors which do this process. Capacitation, and this is first washing of cholesterol, cholesterol vesicles which covering the sperm, then sperm head actually. Then second, removal of membrane cholesterol. As you know, I am talking about plasma membrane actually. Removal of membrane cholesterol. Membrane cholesterol which is present over the acrosome. Then and what will happen after this? This weakens the membrane covering. Weakens the membrane covering. Then fourth point is entry of calcium ion. It is very much important. In next chapter, you will learn more about this entry of calcium ion into sperm. And this change movement of sperm tail from slower undulating movement to rapid whiplash motion like this. You can see the video of swarming of sperm, and so from undulating you can see like this, and then whiplash like this. It's too fast movement, so tail movement uh, converted into undulating to whiplash into sperm with flash motion, and this increases permeability of the weakened ectosomal covering by calcium ion. So next is fusion of gametes. A sperm which has reached the ampullary ischemic junction, part of fallopian tube or oviduct, comes in contact with egg or ovum. Sperm come in contact with egg, and the various events happened to this. Once the sperm and egg meet, there are various events occur, like first is, if you remember the structure of sperm, you can see the outermost part is the ectosome, and that's why the first is the ectosomal reaction. In contact with the surface of egg covering, the ectosome releases its uh, hydrolytic enzymes. This also I explained earlier, and this hydrolytic enzyme is known as sperm lysins. So this is known as acrosomal reaction. There are so many enzymes like hyaluronidase, which dissolves a cementing layer of corona cells, dissolves cementing substance of corona cells. So corona, I think you all remember the structure. Corona cells. It is present in an ovum, and then corona penetrating enzyme brings about corona penetrating enzyme bring about degeneration of corona cells. Degeneration of corona cells. So as the sperm head reaches the zona pellucida. Zona pellucida. So there occur reaction which result in emptying of acrosome and degeneration of zona pellucida by acrosin. This zona pellucida layer is also get degenerated by the enzyme acrosin. So there occur compatibility reaction. Compatibility reaction. Compatibility reaction, and it also stimulate development of an outgrowth by the oocytes, and this is called as cone of reception. Cone of reception. So there is another event, which is first one is ectosomal reaction in fusion of gametes, and then how the sperm enters, sperm entry. You can go through one diagram which you can see here. This is sperm lysine, cone of reception. This is formed and fusion of plasma membrane, corona radiata, zona pellucida layer, plasma membrane, and then this is sperm chromosome, and this is ovum chromosome or egg chromosome, and this is the centriole which facilitate the cell division reaction, and these are the polar bodies which will degenerate further, and this is the cleavage spindle. So this is the stages of fertilization. So in sperm entry, what will happen? A sperm head comes in contact with the fertilization cone. Sperm head plus cone of conception. So what will happen? It triggers a depolarization of membrane, and sperm and egg membrane dissolves in the region of contact. So after dissolution, what will happen? Dissolve, and then what will happen? The head nucleus, neck and middle piece of sperm enter the cytoplasm of the egg. Head plus middle piece, neck, it enters in the cytoplasm of the egg. But tail is left out and it will degenerate further. Tail is left out. Then there occur reaction which is known as actually cortical reaction. So calcium ion is very much important in this. Calcium ion wave causes cortical extrusion of cortical granules out of the egg, and it brings about the modification of plasma membrane and change the same into fertilization membrane. Then next is zona pellucida reaction. This is also called as zona reaction. So chemical which is extruded from the egg act on zona pellucida and destroy the sperm receptor. Okay, this one is very much important because you can see that million of sperms are released by the single ejaculation, but only one gets entry, or only one is able to fuse the ovum, and this is due to this zona reaction, because the chemical extruded from the egg act on this zona pellucida uh, layer and destroy the sperm, destroy the sperm receptors. It means that now none of the sperm is allowed to enter. So it is it able to prevent attachment of any other sperm, any other sperm. Then next is the activation of ovum. So it requires some protein. So prior to fertilization, egg in the secondary oocyte stage in which meiosis second has started but arrested in metaphase second, as I explained you in oogenesis. So there were MPF, that is metaphase promoting factor, and that's why this oocyte, secondary oocyte, was arrested in metaphase second. But the factor has been removed by the APC, that is anaphase promoting complex, which develops and which result in completion of meiosis second. And now the completion of meiosis second, as I told you in oogenesis, that once the sperm is reached there, then this MPF has been removed by this APC and the meiosis second will be completed. So this convert the oocyte into ovum. Now we can call this secondary oocyte into ovum. Actually, from beginning, I'm saying that this is ovum, but technically now this is called as ovum when this completed their meiosis second. So the next step is karyogamy. It is a final stage of fertilization, and the sperm nucleus and the ovum nucleus, it fuses, their nuclear envelope breakdown, and the spindle is formed. Spindle is formed. As you have seen in the sperm structure, there is presence of centriole. So centriole centriole facilitate cell division, and you know that ovum is larger in size and full of cytoplasm, so it provide nutrition to the whole process which is going on. So the paternal and maternal chromosome mix up and resulting in the formation of a diploid set. Paternal plus maternal chromosome mix up and it form zygote. So this diploid zygote then undergo immediate cleavage and will form embryo. So this will I explained further. So this is all about the fertilization and implantation. So fertilization, there are so many topics like arrival of ovum, insemination, then motility of sperm, then capacitation of sperm, then uh, capacitation, fusion of gametes. This also has so many phases: acrosomal reaction and sperm entry, zona reaction, and activation of ovum, and the last one is the karyogamy. So this is all about the fertilization.